Hi there, welcome to my presentation. My topics are Rift Valley fever, Marburg, and Ebola. Let's begin with Rift Valley fever on slide 2. Rift Valley fever, also abbreviated as RVF, is an acute viral hemorrhagic fever that is mainly found in domesticated animals such as cattle, goats, camels, and sheep. They can also cause illness in people. The disease is caused by the RVF virus, also abbreviated as RVFV, which is a member of the genus Flebovirus in the order of Bunia virilis, and it was first reported in livestock by vet officers in the Kenya Rift Valley in the early 1910s. One thing that I've noticed is that viruses are either named by the place where they are found or after the person that first discovered them. So in this case, it was found in the Rift Valley of Kenya, and hence why it's called the Rift Valley virus. And an outbreak caused by RVF can have major societal impact due to economic losses and trade reductions, especially in the livestock and meat industries, because mainly it's the animals that are first impacted. The most notable RVF episodic outbreak occurred in Kenya in the years 1950 to 1951, which resulted in the death of approximately 100,000 sheep. So you can see how that can be detrimental to the economy. Having 100,000 sheep dying would also impact people because there won't be, as much sources of meat for the local population. So the episodic RVF outbreaks increase the likelihood of contact between infected animals and humans, which obviously cause the outbreaks in people. And not only in Africa, but also in Egypt, it was reported that imported domesticated animals which were infected caused a large outbreak that resulted in 600 human deaths. Another outbreak occurred in West Africa in 1987 during the Senegal River Project, which caused flooding in the lower Senegal River area. And that changed the ecological conditions and interactions between animals and the people. And this resulted in a large outbreak, RVF outbreak, that impacted both people and animals. So on slide 3, we are going to discuss the mode of transmission of RVF. So again, RVF is, a zoonotic virus, meaning that it starts from animals. Mainly, mosquitoes act as the reservoir and vector for the virus. They can hold the virus for a long period of time, and then they transmit it to their offsprings when they lay eggs. There are also other insects that are involved, such as arthropods, sandflies, ticks. Those can also act as vectors, depending on the location. So after periods of heavy rainfall, so once the eggs are laid in some body of water, then rainfall occurs and flooding, and that stimulates the eggs to hatch. And once the eggs hatch, those new baby mosquitoes contain the virus, and they go on and bite people, and hence either before biting people, they tend to infect animals by biting them, which is the first point of contact. And once they bite the animals get infected, then people interact with these animals. They either take care of them in the homes or simply eat them and hence get infected. So on slide 3, RVF is approximately 90 to 110 nanometers in diameter. It is an enveloped virus with a single-stranded RNA genome in 3 segments. The 3 genome segments are circular and classified as large, medium, and small. And the RVF invades host cells by endocytosis, as you can see on the right side of the slide. And after endocytosis, the virus replicates via the endoplasmic reticulum and Golgi apparatus, and then once it gets into the cell, it basically goes through the primary transcription and then translation of proteins through the endoplasmic reticulum and then the Golgi apparatus, and basically produces copies of itself and then exits the cell eventually. So in the body, macrophages and hepatocytes and endothelial cells are the target cells in the human host. And these macrophages are mainly involved in recycling, attacking foreign entities. So when these cells are the first target in the body, that causes a reduction in the immunity of that body that has been attacked. On the next slide, I'm going to discuss the signs and symptoms of the disease. RVF has an incubation period of 2 to 6 days following exposure to the virus. It can cause several syndromes when the symptoms do appear in the body. Some infected patients have no symptoms or have mild illnesses, including fever, weakness, back pain. Typically, patients recover within 2 days to 1 week after symptoms start. However, an 8 to 10% of infected people develop more severe symptoms and get diseases such as ocular disease. Some patients report blurred vision and even decreased vision. Some get better 10 to 12 weeks. Others with lesions at the macula, which is the center of the retina. And those people in critical conditions end up with permanent vision loss. Encephalitis or inflammation of the brain also occurs, and that can lead to severe headaches, seizures, and even coma. This occurs in less than 1% of the patients that have the RVF virus. And it occurs 1 to 4 weeks after the first symptoms have appeared. There's also hemorrhagic fever, but it is mainly rare. It occurs in less than 1% of all the RVF patients. The symptoms of this fever may begin with jaundice or other signs of liver impairment. It is also followed by vomiting blood, having bloody stools, bleeding from the gums, the skin, the nose. The symptoms appear 2 to 4 days after the onset of the illness. Fatality is around 50%, and death usually occurs 3 to 6 days after the symptoms start. In animals, RVF causes fever, weakness, and stillbirth, like abortions, like loss of pregnancy, especially in the young animals. So the diagnosis for RVF requires lab testing for both blood or tissue samples. The virus can be detected in blood during illness and in post-mortem tissue by isolating the virus from the cell cultures and performing molecular techniques such as reverse transcriptase polymerase chain reaction. The antibody testing uses enzyme-linked immunoassay, also known as ELISA, which can be used to confirm that RVF virus infection has occurred by showing the IgM antibodies which appear as a response to a recent infection. IgG antibodies also uh, can also show up after several years. Um, After several years, when you notice the IgG antibodies, it's a sign that that person has been fighting the RVF virus. So both antibodies, both the IgM and the IgG, are antibodies are specific to the RVF virus. So far, there's no FDA approved treatments for RVF because most cases are mild and self-limiting. Not specific, no specific treatment has been established yet, and there's no vaccine currently available either. Symptoms such as fever and body aches can be treated with over-the-counter medications. Most people feel better within 2 days to a week time frame. Even though there's no vaccine for people, there's an animal vaccine called the Smith-Burn vaccine, which has been widely used in Africa to control animal RVF. But it is known to cause birth defects in animals and stillbirth, and may only provide limited protection, particularly in cattle. Alright, so that's it for the RVF virus. Our next topic is Marburg. The Marburg virus is a rare but severe hemorrhagic fever that causes fever that affects both humans and non-human primates. And these non-human primates include monkeys, gorillas, chimpanzees. It is caused by the Marburg virus, and it's a zoonotic RNA virus from the Filoriidae family. It was first discovered in 1967 when outbreaks of hemorrhagic fever occurred in Marburg, and that's in Frankfurt, Germany, and also in Yugoslavia, which is present-day Serbia. So what happened was that 31 lab workers became ill initially, and then medical staff got infected. Then family members of the patients got infected, and 7 deaths were reported. The first infected got exposed to imported African green monkeys and their tissues while research was being conducted. The main reservoir host of Marburg is the African fruit bat. Primates, including humans, can become infected and develop serious illnesses with high mortality. So the disease is known as Marburg disease, or it's also called Marburg hemorrhagic fever or African hemorrhagic fever and green monkey disease as well. On the next slide, we discuss the transmission of the Marburg virus. It appears in sporadic outbreaks throughout Africa. Lab-confirmed cases have been reported in Uganda, Zimbabwe, the Republic Democratic of Congo, Kenya, and South Africa. Some of the outbreaks started with male mine workers working in mines that are infested with bat. The virus was then transmitted within their communities through cultural practices and underprotected family care settings and underprotected healthcare workers. So basically, the mine workers would work in the mines and they would get bitten by the bats and get infected, and then eventually they would go, interact with their families, who would also get infected, and the healthcare workers who are underprotected would try to heal them, but they would end up getting an infection. There's been cases that occurred outside of Africa, such as the 1967 outbreak. In 2008, a Dutch tourist developed Marburg after returning back to the Netherlands from Uganda. And also, there was also an American that also visited the same place, but that American recovered from their illness. The Dutch person did not actually, however, recover. The person basically died. But they all visited the same cave. It's a well-known cave inhabited by fruit bats in a national park in Uganda. So hence why people knew that the virus was coming from the bats in Africa. It is believed that their infections occurred via unprotected contact with the infected bat feces, or through the air with aerosols. Then person-to-person infection occurred through direct contact to droplets of body fluids or contaminated equipment which had infectious blood. So on this slide, we see the pathogenesis of the Marburg virus in humans. The first point of contact here is with bats, either being bitten by a bat or interacting with bat feces which contain the virus. Then the body gets impacted by the virus. The immune cells become infected. In the lymph nodes, the immune cells get infected. There's a series of other reactions that occur throughout the body. There's pulmonary edema. There's the spleen gets affected, the pancreas, the kidneys, and it also leads to secretary diarrhea. So we see how hepatocytes get impacted after the virus attacks the body in the liver. And then the lymphocytes also get damaged once attacked with the virus. The kidneys get epithelial necrosis. The blood vessels also get fibrin thrombosis, which creates problems in the circulatory system. After 5 to 10 days of incubation period, the symptom onset is sudden, and then it is marked by fever, chills, headache. Around the 5th day, a maculopolar rash occurs, usually in the trunk area of the body, around the chest, the stomach, and the back. Nausea, vomiting, chest pain, sore throat, abdominal pain also occurs. The symptoms increase over time in severity and can include jaundice, inflammation of the pancreas. There's also severe weight loss, delirium, shock, liver failure, massive hemorrhage, and multi-organ dysfunction. Clinical diagnosis can be difficult because the symptoms of Marburg are very similar with other major infectious diseases. So sometimes it can be mistaken for another disease. The case fatality rate for Marburg is between 23 to 90%. Patients with Marburg must be isolated when infected. They must be quarantined. Health professionals must be notified as soon as possible to collect samples and perform tests to confirm the infection. Antigen capture enzyme linked immunosorbent assay, also known as ELISA, is used for testing. PCR is also used. IgM capture is also used to confirm that Marburg has occurred, and usually these tests are done within a few days of symptom onset to really get a good result. IgG capture is also used to test people who are late in the course of the disease. So for someone who didn't get a chance to get tested initially, an IgG capture can be done to check whether they have received the Marburg virus. There's no specific treatment so far for Marburg. Supportive hospital therapy is mostly utilized, and it includes balancing the patient's fluids, body fluids, maintaining oxygen levels, replacing blood and clotting factors. Alright, so that was it for Marburg. Our next subject is Ebola. The Ebola virus disease is a deadly disease that has recurring outbreaks, mostly in the African continent and also some parts of Asia. It affects people and non-human primates, including gorillas, chimpanzees, and monkeys. Scientists don't exactly know the source of the virus. However, it was first discovered in 1976 near the Ebola River in the Republic Democratic of Congo. They believe that it is a zoonotic disease which begins from animals, and in this case, the reservoir animal is bats, and also non-human primates. The virus spreads through direct contact with infected blood, body fluids, and tissues of the animal. Then it spreads within groups of individuals through the same process, as well as sexual contact. And this happens even if a person has recovered from Ebola, they can still transmit the disease to another person because the virus is very persistent and it can survive. It tries to preserve itself in body fluids, such as the semen, for example, just to ensure that it keeps being reproduced and keep doing what it what it basically like to do. The signs and symptoms appear from 2 to 21 days after the first contact with the virus. The illness goes from dry symptoms like fevers, aches, fatigue, and then to wet symptoms, which include diarrhea, vomiting, and bleeding. Um, Other symptoms include red eyes, having skin rash, hiccups, muscle and joint pain, sore throat, loss of appetite, um, gastrointestinal pain, hemorrhaging, and bruising. The transmission of the virus again starts with bats. There's evidence that suggests that bats are the reservoir of the virus. They carry the virus and they can transmit it to other animals, like apes, monkeys, and antelopes as the bats interact with the environment. So a bat can try to eat a fruit, for example, and that is unfinished, and the fruit can fall from the branch all the way down on the ground, and a monkey can come and try to finish the fruit off and get the virus that way. And then there's spillover events that occur when the animal and humans become infected with Ebola through contact with the reservoir host. This contact could occur through hunting or preparing the animal's meat for eating. So the local population nearby goes hunting in the forest, and they end up killing the animal that was infected by the bat. And when they come home, they try to cook it, or even when dealing with it, when touching the body fluid of that animal, they end up getting infected with Ebola. And the transmission keeps on going. Once the Ebola virus has infected the first human, transmission of the virus from one human to another can occur through contact with the blood and body fluids of sick people, or also those people who have died from the virus. So not only can people get it from infected people, but when they are dead and funeral practices occur, if they don't practice those funerals properly, they can end up getting infected from it. As we can see on the slide, there's also healthcare workers that are also at risk if they are not properly protected. And those who actually survive from the Ebola infection, after they recover, there's still other effects. They report being tired and having muscle ache, and they face stigma, into re-entering into their communities, because everybody's now careful, trying to be careful, and try to basically distance themselves as much as they can, because of fear of not having the virus themselves. So they deal with a lot of stigma in their communities. There are currently no two treatments approved by the FDA for the Ebola virus disease. The first drug is called Inmazeb. It was approved in October 2020, and it's a combination of three monoclonal antibodies. The second drug is Ibenga, a single monoclonal antibody approved in December 2020. Monoclonal antibodies are proteins produced in the lab that act as natural antibodies to stop a germ, such as a virus, from replicating after it has infected a person. So what they do is that they bind to the virus glycoprotein, which prevent the virus from entering a person's cells. Supportive care must always be provided, whether or not other treatments or vaccines are available. And these supportive treatments include providing fluids and electrolytes orally or intravenously to a person that's infected, as you can see here on the right side of the slide. Medications should also be provided to support blood pressure, help reduce vomiting, control diarrhea, and pain, to help with pain as well. So these are the references. That was it. Thank you so much for your time, and you have a good one.